weeks, it's been a recurrent sight across much of Europe, crops peeping through a blanket of snow and ice. Leeks on this farm, close to the French city of Lyon, have been covered for several days, and damage is now unavoidable.
The big freeze gripping much of the nation, especially the East Coast, including Florida. And it couldn't have come at a worse time for citrus growers.
Well, that hard freeze on the North Shore is causing concern for farmers today. WDSU North Shore Bureau reporter Heath Allen is live now. And Heath, how nervous are they really tonight?
Well, I tell you what, they're very nervous tonight. We're talking about, below freezing temperatures, and that's something a strawberry farmer really is fearful of, because the number of hours spent below freezing really threatens that strawberry crop. And so they've gone to
Okay. Hi, we are from Mexico. We are the team from a very cool E. coli. So I hope to enjoy our presentation. We can find it again. It's okay. Sorry, we have maybe to do this here.
Hi.
Ahí déjala.
Sorry.
Sácala de FS. Yo las paso. Yeah. Okay.
Well, every year we witness negative effects on agriculture due to frost. Next, please. Species like cocoa and coffee that are in environments when an average temperatures of 20 degrees Celsius or more are affected by frost, and that causes the temperature to drop to 16 degrees or less. Ice formation doesn't allow gas exchange in plants. In Mexico, the crop that is the most affected is coffee. The agriculture is affected directly by frost. Due to these low temperatures, the crops are damaged, and it also affects the grass that is eaten by the livestock. The damage of the crops not only imply an economical loss, but also cause famine due to the frost. And this can affect the population that depends on those crops as a source of food. Next. Well, currently we know that life can exist under extreme environmental conditions, and such conditions represent a challenge that evolution has overcome. And life has evolved toward cold environments, and these represent almost 91% of the biosphere. Next. Well, to find solutions for the current problem, we went and looked at nature's solution. To avoid this problem, some organisms have developed different strategies, like tardigrades, some fungi, among others, use sugar, especially the disaccharide cellulose. Next, we have the ribosome that works at low temperatures, and then have the thermogeny of the birds that release energy. And last but not least, we have the antifreeze proteins, AFPs. No? This last protein prevent interact with water molecules and inhibiting crystallization. This protein is a consensus sequence of the fish belonging to Suarez genus, and this protein is the nature at a low temperature, but this can be an advantage in its use. Well, we propose a biosynthetic construction that allows E. coli to produce an AFB from 50 degrees. And this protein prevents crystallization in a cell, and it allows its survival at low temperatures. Also, we have developed a switch by using a cold shock promoter, a promoter of a protein that is found in E. coli, which regulates the expression at low temperatures. The use of the antifreeze protein and its regulatory system can help crops avoiding any possible harm by frost. And there are other possible important applications like preservation of tissues and organs, low temperatures, but without harm.
Thank you, Edie. I will talk about our work lab. We built 4 new biobricks from natural sequences. These biobricks have been designed for different responses to cold shock. First of them is a promoter. The promoter consists of 15 pairs of bases and has a rich region of 80, and this is very, very stable from 37 degrees to 15 degrees, and has a rich region of 80, and it confers high stability to low temperature. Then we have the cold box. The cold box may form a steam loop and is very, very important to auto-regulation of the system. Then we have the UTR region. The UTR region consists of 153 pairs of bases and is very stable to 37 degrees and unstable to 15 degrees and very stable to 15 degrees. And the average of half-life to 15 degrees, more or less 20 minutes. Then we have the Schein-Delgarno region, and finally the DB. The DB consists purports too much stability with the ribosome. Nature has too much strategies to survive a low temperature. One of them is the synthesis of antifreeze proteins. Why do we use a consensus sequence of this fish? Because it's very stable to low temperature and is short, only has 63 pairs of bases. That confers too much properties to work at lab. These are our parts that we are proposed for the registry this year. First, the promoter, then our cassette or construction, then from the call box to DB, and then from the call box to Schein-Delgarno. Our purpose is that the users can do different combinations between them. There are reports where have changed the promoter for other kind of promoter constitutive. This and the system works. These tell us that the system is very, very conservative at nature, and the people can work with them and do different combinations, and works. In progress, we have to couple a GPP. We try to do different kinds of expressions of our system, not only one. In this time, we want to probe the promoter and the complete system from our construction from the promoter to DB. In progress, we have this section, and the next step is to prove our system with the promoter and our construction with the GPP. We have to couple this system, our system of response cold shock, and the GPP, the antifreeze protein. At future, we try to synthesize and purify the protein. We think that these have too much application, not only for the, like a protection, maybe we can use a different kind of condition of temperature and try to apply at crops, teachers, and maybe organs conservation. We think that, finally, I want to say that our system is friendly with nature, and we are not releasing organisms, neither DNA.
Hi, I'm Luis, and I'm going to talk about the modeling section. What we try to represent in our model are several characteristics of the cold shock response system. The first one is the cold stress response in E. coli to cold conditions. The second one is the activity of the CSPA. It's the major cold shock protein in E. coli. The third one is the cold shock protein A activity as an RNA chaperone. And finally, the cold shock dynamics. I mean, if bacteria are at 37 degrees and growing exponentially, and suddenly a cold shock is induced, what we expect to see is a decrease in the rate of synthesis of almost all of the E. coli proteins. And the cold-induced proteins like CSPA will reach certain level. After a couple of hours, it is reported that the cell becomes adapted to cold conditions by the aid of the cold-induced proteins, and the cold-induced proteins become repressed. We try to represent all the possible interactions of the system in a big model, but we realize, as always in biology, that the amount of interactions was very high. So we try only to simplify, and we try to simplify the system and only have the more essential characteristics of the cold shock response system. And we did an extra assumption. We see the cold as a molecular agent more than a physical effect. And with this assumption, we implement the model in differential equations in Cell Designer. What we obtain in our preliminary results is that if we have in time zero the cold shock of the, given to the bacteria, and in grid line we represent the amount of all the E. coli proteins, and with the red one, the amount of the CSPA protein. What we can see is that in time after the cold shock, the concentration of almost of the E. coli proteins decreases, but sometime later, when a cold shock protein A has reached a certain level, the synthesis and the amount of the E. coli proteins will reach a certain level. This is happening in our simulation to a wide range of parameters, and this is only the preliminary results that represent only the qualitative behavior of the system. As an extension, we try to fit the model with real parameters obtained from the experimental data, and try to prove that our model is correct and represents the system well. Good. Now I'm going to talk about the collaboration section that we did with our brother team of the same university, UNAM Genomics. What we try to do is a model of an application of the light emission reception system that they developed this year, and have all the genes that they designed in a cycle of 3 nodes with positive feedback between them. We propose a non-linear model where the dependence of the previous node is clear. We have a saturation term and a degradation one. Almost of the equations are the same, and we obtained this. We can see that in the early stages of the simulations, we obtain oscillatory behavior, but the system goes to a stable point, to the equilibrium or to the trivial one, fixed point. So what we are going to do is fit the model again with experimental data and try to prove if we can obtain oscillations, and if not, we will try to prove it formally in a mathematical way. I'm going to finish my part talking to you about the first Mexican workshop in synthetic biology. This workshop was organized by our team and faculty from Facultad de Ciencias UNAM in Mexico and CINVESTAV Irapuato, Mexico. We had more than 90 applications this year, and finally we had only 29 undergraduates from chemists to mathematicians and 7 graduate instructors. We had several activities in the workshop. Among them are brainstorming of short synthetic biology projects, wet lab implementation of them, mathematical modeling of the biological circuits that students built, designed, discussion of human practices, ethical issues, and biotechnology-based business. We had a series of lectures by invited speakers, and the presentation and discussion of all the projects at the end of summer. And by this way, Mexico is part of the first first Latin American teams that promote synthetic biology between undergraduates and gives them the possibility to do foundational and applied research in the field.
Hi, I'm Christian. I'm the crazy guy who the mental assignment let me go to present here at igem. The other part of the collaboration was we had a lot of problems. That's why I'm crazy, because at the beginning of the project, we found problems with the patents. You know, patents is very complicated for teams, and I'm very sure that we are going to find more problems, not only one problem. We are going to find more problems next years in igem if we are not sure about patents. So we try to make a survey. We try to ask the people about the patents issues, and legal advice, knowledge about how to patent, plans to patent pieces, and also the first attempt to fix this is the BPA, the Bio-Republic Agreement. So the most important things here is that we have 132 responses from 68 teams, 24 countries, 110 were students, 13 were advisors, and 10 were teachers. So we can explore almost a little bit of the people inside igem. So the most important question we found here was if the patents related, if they consider it necessary to protect their intellectual property with patents, and mostly, almost a little bit more than the 50%, 58% say yes. The other question was if the people knows about the BPA, and this is very important to take in care because only 63% of the people doesn't know about the BPA, and we have to support these initiatives to help us improve our work in igem. So other part that we make is BioBrick Tools. It's a small automated integrated tool. It's open source based. It's BioJava library. It's extendable. Anyone can download the source code and can extend it. It's BioBrick oriented, and it's fairly community available. So I want to make an invitation to all the people here. If you want to participate in this project to improve for later years, you are free to contact us. So what's the achievements during this igem for us? 4 natural CSP parts we have now in our lab, 1 sequence of AVP that we are going to test. We have a mathematical model. We have simulations of this AVP was modeling in our computers. Also our software BioBrick tools. Again, our research about patents and BPA. We have our first national synthetic biology workshop, and we are part of the first network in synthetic biology with working with enterprise. So here is the people working with us. Thanks for coming. So what's for the future? We have to improve the function of our CSPs. We have to test the AVP. And we have to feed the model with real parameters from lab. And we have to develop a biosecurity mechanism so we can release this protein without harm the medium. So I would like to thank the people here. Sorry about the presentation. Sometimes software goes crazy like me. Any questions?
So, AFP, you're going to have to produce a lot of this, I assume, and you're going to have to purify it, and it's a protein that's pretty cysteine rich. So I don't know if you've thought about techniques or strategies you can use in the future to help in the purification process to make it more efficient.
Okay. Yeah, we were thinking because, you know, when you try to purify the protein, it's very difficult. We want to find mechanisms that we can purify. That's why we use AVP, because there is one stage in the protein synthesized by the cell stops. So only the proteins under the CSP promoter start to produce. So it will be more easy for us to only blend it and then purify the protein from the media.
I have a question regarding your planned application of that of this cold shock or of those cold protein. Are you planning to transfect cells, or are you just trying to spray plants with it? How is the application to crops, and how do you prevent it from freezing that way?
Our plan consists of basically to do. We have thinking on that question, very good question. We think that, imagine that you have a spray protein on the tissue, and you can confer a protection. That's the plan. We don't think to incorporate the genome more to do like a protection and outside the tissues.
Okay, maybe you can refresh my memory on that one. But I thought the cold shock, these cold protection proteins work in a way that they prevent the water from crystallizing, right?
Mm-hmm.
And that way you don't have the harms of the crystallization effect of water crystals within the plant, right? If you spray them onto the plant, it will prevent it from forming crystals, which is an energy-producing process, right? And so it will be liquid water, and the temperature that you're actually having outside of the crop will reach into the cell way more easily than it would be without your protein. It actually would have, there's a danger that it could have a negative effect on your cold resistance of the plants. As I know, there are some strategies where the people spray bacteria, for instance, on crops, for instance, which actually freeze, and the energy that is released by that freezing process outside of the plants protects the crops. And I don't know how that works with your application process.
What we are planning to do is apply it to coffee, and in leaf only. I mean, the fruit is not affected. What is affected is the plant. We expect that the flavor and the properties of coffee are not going to be modified by the protein. I don't know if it answered your question.
The cold shock protein biobricks you submitted, were you able to show that they functioned in terms of turning on under cold conditions?
We had several problems with customs, and our DNA arrived too late. We only obtained the biobricks. We were not able to test test them, but we expect that how we isolated from the equal strengths that are reported in the literature, we expect that they fit with the behavior of the culture system well. And we don't expect that, yeah. We had several problems too, because the cold shock response system is at 15 degrees, and almost all the protocols to express proteins are at 37. So we need to do our research to the condition at that temperature to express the AH-free protein and work with the cold shock system. And that's another constraint that we faced in the development of the project.
Any questions from the audience?
In time series, there is a cold shock, and later the temperature remains at 15 degrees.
Okay, BioBrick G Tools, it was an attempt to make a software to put all the tools we have together. You know, unfortunately for bioinformaticians, we have to take many tools outside, and we have to do the work with all these tools, and it's not integrated first. Also, we have problems with the open source things because you have to pay to many of these software to do the things we have to do. Also, the extendable thing. So anyone can use this software to extend it. You know, I was working with the people with Freiburg also to extend this using the library of BioJava. It's actually to the community. I wanted to release this to the community freely. So it's I want to be. This has to be improved also. It's a small tool yet now. It's only BioBrick request for call 10 compatible, but it can be done the idea is for the next year maybe do this as just like ID programming, just like Visual Basic or something.
Okay, well, thank you very much again.